Hello, everyone. Thank you for joining today's webinar. This is the 9th event from our Sterile Filling 101 webinar series. And today we'll discuss the ABCs of formulation development for parenteral drug manufacturing. The takeaways from today's event will include what you need to know about your formulation before contracting for fill finish, how to determine equipment requirements for your formulation, and studies your CMO can perform to optimize your formulation. Our speakers will also cover a brief overview of what formulation development looks like, how long it can take, and what to expect during the process. Today from Berkshire Sterile, we have Vice President of Technical Operations, Dr. Shufeng Sun, and Associate Director of Formulation and Tech Transfer, Dr. Saran Malhi, to speak. My name is Sarah, and I will be your host for today. Very quickly, we welcome your questions during this presentation. To ask a question, select the arrow in the toolbar at the right-hand side of your screen. Write in your question and click send. So now let's get started. So I will first invite Shufeng to speak. Shufeng, can you first describe what the process of developing a formulation looks like?
Hi, Sarah. Of course. Today we are going to cover the general process of developing a sterile formulation for GMP manufacturing. A sponsor with a new drug product will have already performed some level of formulation development during R&D for tox studies and preclinical studies. Once a sponsor has demonstrated that their product shows promise in preclinical studies and they are ready to move to humans, we need to develop a more robust production process and drug product formulation to ensure we meet their target profile and develop a formulation process that is manufacturable, scalable, and robust. There are two kinds of formulation development work that takes place when a sponsor is preparing their drug product for first-in-human studies. The first is to address any known issues, such as stability, by optimizing formulation composition. For example, if the drug product is expected to be freeze-dried to extend shelf life, but the product was never freeze-dried before, then the formulation composition will need to be optimized to make it suitable for a lyophilization process. Bulking agent, lyoprotectant, or buffer may be required to be included in the formulation. The second is to develop a formulation process without changing formulation composition. In the second case, formulation development for manufacturability is performed. This is where the drug product formulation process is determined to meet the target profile using equipment and processes in line with GMP manufacturing and to meet the desired shelf life of the drug. An example will be that the sponsor has an existing lyophilization cycle, and the development will focus on cycle optimization and scale-up. To begin formulation development, we need to know your target product profile, what the clinical phase you are going into, your preferred container closure, if it is vial or syringe, for example, and your targeted cGMP batch size. These information will help guide us in developing a formulation and a process that will meet your needs and will be manufacturable and scalable. A target quality profile is a set of necessary attributes that the final drug product must have for the product to be used, and the ranges are typically based on the route of administration. For example, the acceptable pH range, osmolarity, and API concentration, among other aspects, are selected to maximize comfort for the patient that is being administered while ensuring efficacy and maintaining safety. An injection product that closely aligns with both the chemical and physical characteristics of the target site in the body minimizes patient discomfort during administration. To illustrate, pharmaceutical formulations should meet a set specification of pH and osmolarity. When designing a drug product formulation, it is best to keep the formulation as simple as possible. In the ideal case, the drug product would involve just the active pharmaceutical ingredient dissolved in water and meet all requirements laid out in the target profile of the drug. However, this typically does not happen. That is when we add excipients to reach the target profile. Unfortunately, by addressing one quality aspect of the drug product, it can inadvertently impact another aspect of the formulation. For example, if we need to raise the pH to align with the target profile, this adjustment may decrease the solubility of the drug product. As a consequence, achieving the desired API concentration becomes a challenge due to this pH adjustment. Formulation development is a balancing act. As we add excipients and adjust the process, we anticipate that these changes will have ripple effects, impacting various facets of the product. Our job is to address each challenge as they arise, working through each until we arrive at a drug product formulation that meets all necessary quality attributes and other criteria. There are 4 critical properties that we optimize for formulation development in general. Let's discuss what these are and how we can adjust the drug product formulation to hit these 4 conditions that we see in nearly every drug product target profile. These are sterility, API concentration, which is affected by solubility, osmolarity, and pH. We will discuss how each target is selected and how we can correct for this in formulation development. Sterility. The most critical quality attribute of an injectable product is sterility. A product contaminated by bacteria or endotoxin may harm the patient, causing safety issue. In formulation development, a sterilization strategy need to be developed. Common sterilization strategies include stereofiltration, aseptic formulation, terminal sterilization. Product stability or compatibility need to be evaluated in formulation development to support the selected sterilization process. My colleague Saran will describe these sterilization strategies and how to select them in more detail later. Drug product concentration. For the drug product to be effective, it needs to have the correct concentration. This target is decided by the sponsor, and the greatest challenge in formulation development is solubility. All solvents, including water, has a limit to how much solute it can dissolve. If solubility is low, we are not able to reach the target concentration because the API will begin to precipitate out of the solution before we reach the target concentration. So we adjust the formulation to increase the solubility of the solution to reach the API concentration we need. There are several ways we can do this. We can add surfactants. These are excipients that reduce the surface tension of aqueous solutions when dissolved, and they disrupt the intermolecular forces of water, making it easier for solute molecules to break apart from one another in solution. At high enough concentrations, surfactants will also form micelles that surround hydrophobic substances and allow them to be easily dissolved in water. We can also adjust the pH. The solubility of many compounds is dependent on their ionization state, which can be affected by changes in pH. Lowering the pH can enhance the solubility of basic compounds. while raising the pH can increase solubility of acidic compounds. On the other hand, adjusting the pH to a level where a compound is less ionized or where it forms less soluble salts may decrease their solubility. It is important to understand how pH will affect the solubility of the API. And for most projects, once they reach the formulation development stage for first-in-human studies, this will already have been closely studied, and the sponsor can quickly convey this relationship to their CDMO partner. Otherwise, the CDMO will need to perform an additional study to understand this relationship for their drug substance. Solubilizers or co-solvents are often organic solvents like ethanol or glycerin that enhance API solubility by disrupting intermolecular forces within the solute and reducing cohesion. And finally, complexing agents like cyclodextrin can form inclusion complexes with APIs to encapsulate them and increase their solubility by reducing aggregation. There are other agents that can be used to improve solubility, but these are the most common excipients we can add or adjustments we can make to improve API solubility. Once we have achieved the target concentration, our focus shifts to addressing other critical physical and chemical properties, such as osmolality. The target osmolality is typically chosen to align with that of the injection site. If osmolality of the drug product falls below the desired range, we introduce sodium chloride to elevate it. On the other hand, if osmolality exceeds the target, we explore options such as reducing the concentration of the API and increasing the dose volume. This allows us to decrease osmolality while maintaining the same API load per dose. For example, instead of administering a 1 milliliter injection with an API concentration of 3 mg per ml, we may opt for a 1.5 ml injection with 2 mg per ml of API. The API load remains 3 milligram in both injections, but we can expect a lower osmolality in the second injection. pH. The target profile is also selected to align with the pH of the injection site. As a general guideline, the pH of the drug product should go towards neutral, but this may impact the solubility and the stability of the drug product. The sponsor will typically understand how pH affects solubility of their drug, but if not, then additionally studies may be needed to be performed. To adjust the pH up, we add a base, and to adjust it down, we add an acid. An approved list of acids and bases for pH adjustment in parenteral formulations is readily available. Once we have successfully developed a formulation that aligns with the physical and chemical properties specified in the target profile, the next step in formulation development is to analyze and optimize the product for stability. In this stage, we aim to meet two distinct stability objectives. Short-term stability. We need to ensure that the drug product will remain stable for the time and in the conditions it will face during formulation, filtration, and filling. These conditions are usually controlled room temperature, and this process can last up to a week. If the drug product is exceptionally temperature sensitive, it may be formulated in and filled from a jacketed mixing vessel that is kept at 5 to 15 degrees Celsius. If drug products require frozen storage conditions, it needs to demonstrate reasonable stability during freeze-drying cycles. If they cannot last a week at the conditions, it will experience in production, then manufacturing an effective drug product presents high risk. We are also looking to improve long-term stability. We must assess how long the drug product maintains its quality during long-term storage conditions. If your drug product isn't stable for at least 12 months, you are going to have a difficult time maintaining a continuous supply to patients, which could disrupt the clinical trials. That is why we optimize the drug product solution to meet the target profile, which typically extends beyond 12 months mark, often ranging from 18 to 24 months or more. This extended stability duration ensures a more reliable and a consistent drug supply for patients throughout the clinical trials and beyond. Let's start with discussing how we can evaluate short-term stability. One method involves conducting a hold time study, which involves preparing the drug product formulation and placing it under the expected manufacturing conditions. These conditions typically involve either controlled room temperature or ejected mixing vessel chilled to 5 to 15 degrees Celsius. Samples are then taken throughout the study at regular intervals until 6 days or 144 hours have passed. These samples undergo quality testing and critical parameters are graphed over time to identify if and when they fall out of range. If the formulation remains within acceptable ranges and the target profile is achieved for the full duration of the study, then the drug product formulation is considered stable enough for production. If the formulation falls out of range before the end of the study, then the formulation will likely need to be modified to enhance stability, or the process will need to change to protect the drug product from degradation, either through manipulating the conditions it will experience through the tools we have at hand, like ejected mixing vessels, or by putting reasonable time constraints into the production. Long-term storage is equally significant. Many drug products will have a target profile requiring at least 18 months of stability. Early stability analyses can help identify issues with long-term stability without investing unnecessary time in development, waiting for ICH stability results. Early stability analysis is performed for 3 to 4 months using lab samples that are held at accelerated temperature time points. For example, if your drug product should be stored at the controlled room temperature long-term, then early stability analysis will place samples of your drug product formulation in a 40-degree chamber. Several months later, the samples are pulled and undergo quality testing. When you anticipate more than 12 months of stability of your drug product, it's generally expected that these samples remain within the acceptable ranges, even at the 4 months mark under accelerated conditions. If the results fall out of acceptable range during this accelerated analysis, it suggests that further formulation development is necessary to achieve the target long-term stability. How to improve stability if there's an issue with short-term or long-term stability? We have a few approaches we can take. Adjusting the formulation. We have the flexibility to choose alternative excipients that not only meet the target profile, but also anticipated to improve stability. If we observe pH fluctuations over time, we can adjust the pH of the formulation or use buffering agents to control and maintain a consistent pH level. If oxygen is a problem, we can incorporate antioxidants to protect against oxidative degradation. making processing changes. Sometimes process changes are necessary to truly improve the stability of the drug product. We might explore lyophilization as a stabilization technique. For oxygen-sensitive drug products, we can remove the headspace in the drug product and purge it with stereo nitrogen gas. We may need to implement new process changes to limit the light, shear, or temperature during production or for storage. We have a webinar that discusses these topics in detail. Changing the container closure system. The problem might exist elsewhere, in the product contact materials used in manufacturing or in the container closure system. These can quickly be tested to determine if they are a good fit, and if they are not, then a substitute will be selected. Once the final drug product formulation has been established, ICH stability studies can be performed using samples from an engineering run or GMP batch. ICH accelerated studies are held for at least 6 months at an accelerated temperature unit, and these can provide an initial estimated exploitation date for your drug product lot, while longer ICH nominal studies are in progress. These run for duration outlined in your target profile at the nominal storage temperature for your drug product, and they establish a true expiration date. Today, we covered how we develop and optimize drug product formulations for cGMP manufacturing. The next step after establishing a final formulation is to develop a formulation process at the scale which involves selecting the appropriate equipment. Long will discuss this.
Thank you, Shufeng. I will switch over to Saran now to discuss determining equipment requirements.
Thank you, Sarah. As you said, Shufeng, our initial focus is on developing a formulation that aligns with the target profile and then optimize it for the stability of the product. After that, we shift our attention to scale up the manufacturing process by taking into account any product sensitivities or the challenges that need to be addressed. The scale of the drug product batch has a significant impact on the formulation and sterile filtration. For scale up, we need to select the correct size filter, ensure the process designed for efficient mixing and with efficient time being assigned to each mixing step, and use an appropriate size equipment for the desired batch size. Once we have a process established, we'll perform a scale-up run to ensure the equipment selected works as expected, that we are achieving the target product profile and a quality requirement, and to gain a better understanding of the process so we can prepare the formulation team for the engineering and the GMP batches. I want to spend some time today to describe how we determine equipment requirements for a formulation. Some therapeutics already have some known equipments and requirements. For example, if we are creating a lipid nanoparticle formulation, then we will need special equipment such as microfluidic mixer and a quattroflow pump. If we are working with a high shear sensitive drugs, then we may already suspect that this require a special low shear mixer. However, there are many scenarios where it is not clear yet what equipment will be needed. For example, some of these scenarios include if we are manufacturing a very large scale batch, if we are formulating a very viscous formulation, or if we are making a suspension formulation. It also applies to the formulation if the material is temperature sensitive, light sensitive, shear sensitive, or the material has an incompatibility with the commonly used material in the pharmaceutical manufacturing. In those cases, we may require special equipment and material for manufacturing. If we are not sure which equipment is needed, we'll formulate the drug product in lab using the standard equipment for GMP fill to determine if it's effective. If it's not effective, then we'll try implementing equipment that we know will address the issue, and we are seeing and testing the process again. Equipment selection relies on our expertise and deep understanding on formulation, filtration, and filling challenges, especially for the products that are highly viscous, has suspension-based formulation, and has a common sensitivities. We draw from our expertise to identify the right equipment or adapt to the process. To address the current issue, testing the process for real-world concerns. While the equipment selection process can be intricate, we illustrate by examining specific drug product and how we can pinpoint the right equipment for those projects. For parenteral products, sterilization is a critical step in fill finish. There are three main primary methods for achieving a sterile parenteral formulation. First is terminal sterilization. The second is sterile filtration. And third is sterile filtration of each component and then using an aseptic formulation technique. If your drug product can tolerate terminal sterilization, then you are required to perform this. This is the lowest risk approach for achieving sterility into the parenteral products. However, it is important to acknowledge that majority of the drug products that are not resilient enough to the extensive energy associated with the terminal sterilization process, whether this can be a steam sterilization, gamma, or E-beam. When terminal sterilization isn't feasible, the alternative is sterile filtration. In this process, the product is first formulated and then passed through 2.2 micron filters that eliminate all microorganisms from the formulations. However, the selection of the filter is not a straightforward choice. It involves identification of appropriate size filter and a membrane type that are tailored specific to the characteristics of the formulation. The selection of the filter must be of the correct size. If the filter is too small, then it becomes clogged by the bioburden it is filtering out of the bulk formulation, causing the premature ending of the filtration process, which leads to a significant loss of the drug product. If the filter is too large, it may result into substantial holder volume, causing wasting of the drug product and lead to the lower yield of the process. The material of filter also matters. There are many popular filter types, such as PVDF, PES, cellulose acetate, PTFE, and many more. When developing a filtration process for a new formulation, we filter the drug product formulation using at least 3 different filter membranes and evaluate the flux rate from each filter. The filter with highest flux rate is often selected for a cGMP manufacturing because this filter requires the smallest surface area to filter the drug product, which means that less filter holder will be there. However, it's equally important to verify that API concentration before and after filtration remains similar to ensure that filter is not absorbing the API. Certain formulations are not easily filterable. Typically, these are the formulations which are polymer-based or the formulations with an exceeding high molecular weight or concentration, making it challenging task to pass through the filter effectively. For example, the situation is particularly prevalent in the formulation with a high concentration of hyaluronic acid. After thorough testing involving various filters, membranes, and sizes, and utilization of a pressure vessel for highly viscous product, if it is determined that the product cannot be filtered, the alternate approach will involve the aseptic formulation. Aseptic formulation is performed by sterilizing each excipient and the API, then performing formulation under a grade A environment. This approach is considered as a last resort due to the high risk of the contamination compared to the sterile filtration. The process is also more complex and costly. However, we do have a dedicated formulation isolator for the products that require this approach. Some product may be incompatible with materials that are used in the fill finish. For example, we may have formulations that cannot be in contact with silicon, a common material which is used for tubings in pharmaceutical manufacturing. If we suspect that there is an incompatibility concern, then we must test this in very early in the formulation development phase. This is done by placing a material of choice in contact with formulation. For example, placing a small section of silicon tubing into a formulation and holding for several days, during which samples will be collected at a regular interval and testing for quality attributes to identify any detrimental effect, such as a decrease in the concentration of the drug substance or excipients, change in the visual appearance of the formulation, or change in the impurity profile or the pH of the formulation. If we notice that there is a problem with a specific contact material, then we retest to confirm these results. If it turns up with the negative results, we may perform a dynamic compatibility evaluation where the material is only in contact with the formulation in a moving state. For example, formulation will be recirculated through the material such as tubing or the Y connectors for an interval of an anticipated manufacturing time. Samples will be collected and tested at a regular interval. If test results turns up negative again, then we evaluate the substitutes and undergo similar compatibility evaluation with the new material of choice. Maintaining the homogeneity in suspension. Ensuring homogeneity is a critical factor when formulating and filling suspension, especially during the dosing of each vial or syringe from the bulk drug product suspension. Suspensions are prone to settle when formulated under an unmixed state. Filling those formulations can lead to a concentration variance across the filled units throughout the lot. This means that all filled units at the beginning of the lot may have higher concentration of suspension, while the units which are filled at the end of the process may have lower concentration. Settling down can also have an adverse effect on certain lyophilized products. For these products, settling can impact the quality of the lyophilized cake and can also adversely impact the reconstitution time. Improving homogeneity begins in formulation design. Appropriate excipient and manufacturing process should be selected to maintain desired particle size and uniform distribution of the formulation. This can be achieved by incorporating inhibitors that discourage particle aggregation and hinder the formulation of the large particles. As an initial approach during formulation, filtration, and the filling, the bulk drug product should be kept under a continuous mixing to maintain homogeneity and to prevent particles from settling down. If this approach is not sufficient, then we may consider increasing the viscosity of the formulation, which will affect the slowdown of the settling rate, improve overall homogeneity of the formulation. So let's take an example of a suspension formulation and take an example of a lipid nanoparticle formulation and see what are the tasks which are involved in the optimization of that formulation. Let's break down and understand these tasks one by one. First, we start with the solution preparation. Adjustment to the lipid concentration can be made to encourage the formation of the smaller particles while minimizing the aggregation. It is essential to fine-tune the lipid formulation and APS solution concentration to have sufficient encapsulation efficiency and to ensure that we are capable of achieving the desired particle size. Second step in this optimization is the optimization of the lipid nanoparticle formulation process, where we need to optimize the channel dimension of the microfluidic mixer, and we need to modify the flow rates to achieve the ideal particle size distribution. The next step in the process which need optimization is the tangential flow filtration and the sterile filtration, where we must keep in our mind that lipid nanoparticles are shear sensitive, and shear can destroy the lipid nanoparticle, causing the aggregation and creation of large particles that can widen the particle size distribution and can eventually hinder the sterile filtration. The optimal mixing speed, pumping speed, and the equipment will be needed to be evaluated to ensure that drug product is not being damaged by the shear forces. If they are, we can slow mixing speed and employ special low shear mixing mechanisms and vessel to protect the formulation. Overall, optimizing each of these stages will improve the quality of the formulation. Let's talk about the sensitivities of the formulation and find the right equipment and for the drug product that are sensitive to light, temperature, and shear. We won't go into too depth of this topic, especially because we already have a webinar dedicated to this topic, but we will list out some formulation adjustments and equipment adjustments that can be made for these type of formulations. The first one is temperature sensitivity. This is the most common formulation sensitivity which we see, and there are many ways to manage this concern. In formulation development, it is critical to select excipients that are are capable of improving the stability of the formulations. We can add stabilizers, antioxidant, buffering agents to protect the API degradation, or we can make a pH adjustment to a range that is less prone to the temperature, induce changes into the profile of the API. Once we have done what we can reasonably do in developing a formulation, the stability enhancement options, we can manage temperature sensitivity by keeping the formulation under a cold environment during the formulation and the filling process by employing a jacketed vessel. The additional things which we can do is like we can limit the production time, and we can also store the drug product at an appropriate temperature control unit. If all that is not enough, then we may consider lyophilization as a technique to enhance the shelf life of the finished product. The next sensitivity is the light sensitivity. Some formulations are light sensitive, and in formulation development, we again optimize the excipient and the pH to enhance the stability of the formulation. In manufacturing, we can work under special light conditions such as amber light, or we can cover the formulation in a light barrier to keep it in a dark environment. The drug product will also likely be filled in an amber container and stored in an opaque box after filling to keep it protected from the light exposure. The next sensitivity, which is also a concern, is a shear sensitivity. Shear sensitivity can damage the formulation, especially for the protein formulation where aggregation is a concern. Unfortunately, shear forces are present throughout the manufacturing process, whether it be mixing during the formulation or in a pump action during the filtration and the filling operation. As mentioned earlier, inhibitors can be added to the formulation to limit aggregation, and the excipients and pH should be optimized to enhance the stability of the formulation. Shear testing can be performed by evaluating different mixer or pumps and then identifying the optimal mixing and the pumping speeds for processing these formulation during manufacturing. Once again, all these sensitivities are discussed in detail in an earlier webinar and how they can be evaluated and managed. Let's briefly discuss viscous formulations. Not much can be done to lower the viscosity of the formulation other than by reducing the concentration of the formulation. And many times the viscosity is a critical attribute towards the performance of the finished product. Developing an effective GMP process will mostly depend on selecting an appropriate equipment to handle the challenges for the viscous formulation, filtration, and filling. We created an in-depth webinar on this topic that should explore if it is applicable to your product. But we will cover very quickly what equipment can be employed and when and why these equipment are useful for these formulations. During formulation, we need to effectively mix the product to make it homogeneous. However, be cautious to limit the creation of the bubble while doing so. We will most often employ an overhead mixer and mix the formulation slowly to limit the introduction of these bubbles into the formulation. Most formulation will also have a wetting period. For example, formulations where hyaluronic acid is added, we leave the formulation overnight to allow wetting. Doing so will help us speeding the mixing process for the formulation. During filtration, we may employ a pressure vessel to help push the solution through the tubing and the filter. If we do so and use a pressure vessel, we will likely need to use a pressure-rated tubing, especially on the pre-filtration side of the filtration assembly, to prevent any leakage during the filtration process. Once the solution passes the filter, the pressure drops. So considering that on the post-filtration side, leaking is not a concern and pressure-rated tubing may not be required. During filling, most likely we need to use a piston pump to fill the product. This is a more mechanical process for filling that is more effective for viscous drug products. If the viscosity is significant, then we may also be filling from a pressure vessel to help push the solution through the tubing. And here again, we employ pressure vessel rated tubing both onto the inlet and the outlet side of the pump. The reason we would use a pressure rated tubing on the outlet of the pump is to prevent the tubing from bulbing. When the pump runs, it will push out a lot of product into the outlet of the tubing. We don't want the tubing to bulge or relax over time because this can lead to the dribble of the formulation and can cause the inconsistent weight checks in the filling. If you want to learn more about the viscous filling, I recommend you watch our previous webinar on this topic. So to summarize, formulation development starts when developing a drug product solution that meets the target profile. Then we must create an effective formulation and manufacturing process to formulate at a scale in a GMP environment. Developing an effective formulation process for cGMP manufacturing requires significant work and a deep understanding of the fill-finish process. Specialized equipment, materials, or processes may be necessary for the drug product sensitivities, high viscosity, suspension, or compatibility issues with the pharmaceutical materials. However, the result of all this work will be an effective and optimized formulation and the manufacturing process that is ready for cGMP manufacturing.
Thank you, Saran. That is the end of today's presentation. In just a moment, we will transition to the Q&A portion of the webinar. Again, to ask a question, select the word bubble that appears in the toolbar at the right-hand side of your screen. An area to type in your question will appear. Write us your question and click send. We'll display those for our speakers to answer. Today's webinar is the 9th webinar of this series, Sterile Filling 101. All of our upcoming events are listed here, and recordings of previous webinars are available on our website or YouTube channel. In November, we will be airing our next webinar regarding aseptic filling challenges for intravitreal drug products. Following the end of this event, I will be sending you each an email with a sign-up link to each of these events, but you can also sign up by going to berkshiresterilmanufacturing.com/sterilefilling101, and the event will be listed there for you to register to. Okay, it looks like we've got all our questions, so let's get started with those. This one I'm going to direct to Shufeng. The question is, are there any regulatory requirements that impact drug formulation development?
Yeah, certainly, Sarah. For the drug in U.S. clinical trial and compared to European marketed clinical trial, the regulatory requirement for raw material and the sterilization process are different. So knowing a target market, it would help formulation development to cover those requirements. For example, choosing an excipient that is multi-compendial compliant.
Thank you, Shufeng. Another question that will go to Saran. How long does formulation development take?
Okay, so it all depends upon the complexity of the formulation. So if it's like a simple parenteral liquid formulation, it can take up to like 4 months. And if it's a lyophilized formulation, it can take up to like 6 months, which includes that we will identify a prototype formulation, and a stability evaluation of that prototype formulation will be performed.
Well said, Saran. We're going to go back to Shufeng for this next one. The question is, how do you select the specific excipients used in a formulation?
When you choose a specific excipient, we typically want to choose the one already come from a qualified vendor in our system. This ensures quality and ready supply, and we may already have this in-house. Those are usually preferred. If a novel excipient is required that is not already in our system or has to come from a unqualified vendor, we will have to consider the vendor's credential and quality history in our selection.
Thank you, Shufeng. Back to Saran. What is the minimum number of days of short-term stability that a product needs to be manufacturable?
The solution's stability of the formulation is very important to allow us flexibility in manufacturing operations. So in an ideal situation, 2 days formulation stability is very important to give the flexibility. We have seen cases where we have clients which have 24 hours of formulation stability, which is very common. In some cases, we also handle the processes which have a 12-hour solution stability information. For those cases, we usually apply the off-the-line visual inspection to minimize the time of the manufacturing.
Thank you, Saran. Shufeng, how do you prioritize which physical and chemical properties to target when developing a drug formulation, especially when trade-offs may be necessary?
This trade-off need to be determined with our sponsor. Depending on their clinical route of administration and the drug indication, some property are more critical than others, and we may be able to prioritize one over the other.
Thank you, Shufeng. We have another one for you. Are there any specific considerations for pediatric formulations?
Yeah, pediatric formulation usually require a much more stringent quality criteria. For pediatric patient, their overall body fluid is in a smaller volume, and they can, tolerate a much less toxicity of the drug. So when we choose excipients and choosing product concentration, we need to keep those in mind. And we typically want to look at the excipients that's already approved to be used for pediatric application.
Thank you, Shufeng. Saran, this one will go to you. Are there any notable trends or advancements in personalized medicine that are influencing formulation development?
Yes, we have seen a notable increase in the clients who are working on the personalized medicine. BSM has worked with multiple clients in getting a GMP manufacturing for these personalized medicine done. The biggest concern which we face in this personalized medicine is the availability of less material. So to address that, we have designed a low loss fill that is capable of achieving the desired target for the clinical supplies for these personalized medicine.
Well said. Saran, here's one for you. How do you mitigate the risk of precipitation of poorly soluble drugs?
Poorly soluble drugs are it's a very common concern, and there are multiple approaches that can be used to address this issue. Some of these are using a co-solvent approach, or we can use a surfactant, or we can even use an inclusion complex based formulations to address these concerns.
Thank you, Saran. We're going to end on that question. I want to give a huge thank you to everyone that joined today's webinar, and a thank you to our speakers, Dr. Shufeng Sun and Dr. Saran Malhi. Thank you again for your input in today's event. To all of our viewers that are here today, keep your eye out for my email, and don't forget to sign up for other events, which you can do by going to berkshiresterilmanufacturing.com/sterilefilling101. I hope you took a lot away from today's presentation, and I hope to see you again in our future webinars.